Hi Tuesday, Thursday class. So I know this is a little bit different, but I thought this would be the best way for me to be able to still lecture to you without actually being there. I did post an announcement on Canvas for questions for this lecture specifically, so feel free to ask questions there. You can reference the slide number, which is down here, or you can reference the timestamp of where it's at in the video. And then that way I will answer questions through Canvas. So if anyone else has that same question, or if there's follow-up questions, we can do it that way. But as always, you can always email me or text me or come see me in person if you have questions as well. Okay, so last time we talked about carbohydrates. We looked at aldoses and ketoses and their chain forms and a handful of reactions that involved the carbohydrates. We're still looking at carbohydrates. But what we're going to look at now is the fact that they form rings. So carbohydrates exist in three forms. Here is our carbohydrate in its chain form. So this is D-glucose. With carbon number 5, the alcohol on it can do a nucleophilic attack on carbon number 1. So our carbonyl bond opens up, we're going to do some proton transfers, and that's going to create a ring. Now, carbon number 1 is going to become a hemiacetal. So this is reaction number 7. That's our name up here. And so remember, that's when we have an OR group and an alcohol attached. An acetal would have 2 OR groups attached. This is going to create a new stereocenter when carbon number 1 becomes an alcohol. And that alcohol can point down, and that's what gives us the alpha form of the ring. Or that alcohol can point up, and that's what gives us the beta form of the ring. The rings that we're drawing are called Haworth projections. So we're drawing our cyclohexanes as a flat structure. And the idea is that this front half is bolded because it's coming out at us. Now, when you're actually drawing this on paper, you don't need to do that, but that's what it is representing. And we always draw the oxygen that's part of the ring in that top right corner. So just like when we draw our carbohydrates as chains, there's a very particular strict way to do it. The same is true when we draw them in rings. And also, just like the Fisher projections are really only used for these biochem molecules, the same is true for these Haworth projections. It gives us a really easy way to see subtle differences in the molecules, but they're just not used in a whole lot of other formats. Now, in how or when these rings form, it can be a lot of different ways. So here's my little list here. Carbon 4, 5, or 6, the alcohol on it can cause this attack. Now, how do we know which carbon's going to do it? It's going to depend on how many atoms there are. So that list there, that's for the ketoses and the aldoses. It's for these 6-membered rings, and then on the next slide we're going to look at 5-membered rings. So you would have to be told to draw the 5-membered ring in the alpha form or the 6-membered ring in the beta form, and then you're going to figure out on the chain which carbons are going to cause that cyclization. So when we draw the Haworth, we talked about that we're drawing a 6-membered ring. We're going to put that oxygen in the right-hand corner. A lot of times carbon number 6 right here is going to be sticking off of the ring, so that's always going to be at this position pointing up. And then if you're told to draw the alpha, you're going to point the alcohol on carbon number 1 down. If you're told to draw the beta, alcohol number 1 is going to be pointing up. So that's what's set. That's the stuff we don't have to make decisions on. The rest of it are going to be the alcohol locations or alcohol directions for these 3 carbons. So notice they're color-coded. The alcohols that are on the right-hand side are always going to point down. And the alcohols that are on the left-hand side are always going to point up. So I always like to think of it as that this molecule just fell over, and when it falls over, these are going to point down, and this is going to point up, and that's what helps me remember it. Last thing I think on this slide is that carbon number 1 is called the anomeric carbon. So again, it's just a name for us to be able to reference it. It will come up in later reactions, so it is a name that we need to know. And this is, I know I did mention it, but this is reaction number 7 of our list. So here is our 5-membered ring. So same idea, we're going to cyclize it from one of our alcohols. So here's our carbohydrate. We're going to use the alcohol on carbon number 4 because 1, 2, 3, 4, and that oxygen makes the 5th member of that ring. So here's our basic drawing for the Hayworth for a 5-membered ring. We're going to put the oxygen top center, and then again, this last carbon is going to just be pointing up next to that oxygen. Now this isn't always going to be there. It's just going to depend on how many carbons were in our chain to start with. But if there is an extra carbon, it will be at this position pointing up. And then alpha, alcohol points down, beta, alcohol points up. So these other two positions, because they're both on the right hand side, they're both going to point down. So it's the same set of rules that we had for the 6-membered ring. Of course, they get names. When we have a 6-membered ring, it's called a pyranose ring. A 5-membered ring is called a furanose ring. So we do have a way to name our carbohydrates when they are in these ring forms. We start with alpha or beta, depending on whether that alcohol is pointing up or down. We use D or L to tell us which carbohydrate we were using. And then the carbohydrate name, but we take off the ending ose and replace it with either pyranose or furanose. So there's a lot of options there. But for this ring, we used D-glucose, and it's in the alpha position because our alcohol is pointing down, and it's a 6-membered ring, so it's a pyranose. So when we put all that information together, we get alpha D-glucopyranose. Over here, it's also alpha, it's pointing down. We started with a D-ribose, and it's a 5-membered ring, so it's a furanose ring. And again, we put that all together, we get alpha D-ribofuranose. Now, I did say that we could do this with ketoses as well. And really, the biggest difference here is that we will have this additional carbon group sticking off of the ring, and which carbons we're using is going to change a little bit. So we have D-fructose turning into a furanose and a pyranose ring. So if we count, so I'm going to use different counting than what's on here because we're looking at how many atoms are going to be in our ring. So this is our carbon that's going to get attacked. 1, 2, 3, 4, 5. So if we're making that 5-membered furanose ring, we're going to use the oxygen on carbon number 5. So here's our nucleophilic attack. Our carbonyl opens up. We do some proton transfers. So this is carbon number 5 right here. Carbon number 6 and its alcohol group is going to be pointing up. There's our oxygen. Carbon number 2, this one right here, is our anomeric carbon, so our former carbonyl carbon. So we have that CH2 that was already on the ring. Our alcohol is pointing down because we drew the alpha form of the ring. And then for carbons 3 and 4, carbon 3, the alcohol is on the left, so we point it up. Carbon number 4, the alcohol is on the right, and we point down. So we end up with this as our final structure. Now starting with D-fructose, we can still do a 6-membered ring. 1, 2, 3, 4, 5, and that oxygen gives us the 6th atom in that ring. So we're using the oxygen on carbon number 6 to bond to that carbonyl. Nucleophilic attack, our carbonyl opens up, we do some proton transfers. So we're drawing our Hayworth in the same way, that oxygen's in that top right corner. Because the last carbon in our chain is the one that is using its oxygen to form the ring, there isn't anything else attached to it besides hydrogens. So we don't have that extra group right here. So there's our anomeric carbon. We have that CH2OH, that first carbon in the chain. We have the OH, so this is the oxygen that used to be the carbonyl. And then for oxygens 3, 4, and 5, 3 is on the left and 4 and 5 are on the right. So we end up with 3 pointing up and 4 and 5 pointing down. So again, we're following the same rules, just we're basing it on how many atoms we need in the ring. So let's practice a couple. We are going to draw the alpha furanose of D-arabinose. So let me switch here to a pen, and we're going to start by drawing the basic furanose shape, which means we're going to draw our oxygen and our ring. Now if we count our carbons, we're going to say 1, 2, 3, 4, 5. I know this is 1, 2, 3, 4. So that means 5 is going to be pointing off of the ring. So there is carbon number 5. And I know it's alpha, so that means that right here, carbon number 1 is going to be pointing down. So that means carbons 2 and 3 are the ones we have to decide. So carbon number 2, our alcohol's on the left, so that means it's going to be pointing up. So I'm going to go right here. And carbon number 3, it's pointing to the right, so that means it's going to go down. So there we go. That is the alpha furanose version of D-arabinose. All right, let's do one more. Well, we're going to do a couple more. Let's look at the beta pyranose version of L-xylose. So again, we're going to start with that basic structure, which means that we need to have our Hayworth 6-membered ring. And again, I'm going to number my carbons. I'm going to switch colors this time though. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay, we know that it's the beta version, so we know that our oxygen here is going to point up. That looks like a little mini arrow. It wasn't supposed to. Carbon number 5 is the last carbon in the chain, so that means that there isn't anything else attached to it coming off of the ring. So carbons 2, 3, and 4 need to have their alcohols. So our alcohol on carbon 2 and 4 are both on the left-hand side, so they're both going to point up. And the alcohol on carbon 3 is on the right, it's going to point down. And that's our finished structure. We have all of our alcohols accounted for. So this is the ring structure, or the beta L xylopyranose, there's too many consonants in a row there on that one, form of this carbohydrate. So let's look at a couple ketose versions. So we're going to draw the beta furanose version of D-xylulose. So again, we're going to start with that ring shape. So there's my oxygen and my 5-membered ring. And I'm going to number my carbons here. 1, 2, 3, 4. Now I numbered starting at the carbonyl because that's where my carbonyl is. So 1, 2, 3, 4. How you number it doesn't really matter, just as long as everybody's accounted for. We know we don't want to magically lose a carbon or magically gain a carbon. So as long as you're numbering them and try and be consistent with how you number them, that's what's going to help you out. So because we want to draw the beta version of this, we know that our alcohol is going to be pointing up, but then also attached to carbon number 1 is this group. So we're going to point that down, and it's just pointing down by default, really. The beta tells us that this group points up, so then if there's anything else attached to it, it has to point down. And the opposite would be true if we had the alpha form. So we look at the rest of our structure here. Carbon number 4 is the last carbon in our chain, so it doesn't have any substituents coming off of the ring, but carbons 2 and 3 do. So carbon 2's alcohol is going to be pointing up because it's on the left, and carbon 3 is going to point down because it's pointing to the right. All right, one more. Let's draw the alpha pyranose version of L-psychose. So again, we're going to start with our basic ring shape. And I'm going to number my carbons. And again, I'm going to start at that carbonyl. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. We are drawing the alpha version. So there's my alpha alcohol pointing down. Also attached to carbon number 1 is that CH2OH group. Carbon number 5 is the last one in the chain, so there aren't any substituents coming off of that ring. 2, 3, and 4, all my alcohols are on the left, so that means they're all going to point up. So 4, 3, and 2. So there we go. There's our final structure of our L-alpha psychpyranose. So we're going to look at another way to draw these. We're going to draw the Haworth projections as chairs. So the Haworth projections are definitely the biochem way to do it, but as we know, the organic way to do it is to draw chairs. And so we should be able to take our Haworth projections and redraw them as chairs. And here's our little tips and tricks to do this. We're going to take carbon number 4 and we're going to point it up. So we're treating that as the headrest of our lounge chair. And carbon number 1 is going to point down. So it's going to be treated as the footrest of our chair. The CH2OH group, if there is one, is going to be equatorial because of its size. And then all of the other groups are going to be drawn in relation to it. So whether they're both pointing up or both pointing down, so whether they're cis or trans, to that CH2OH group, we're going to draw them in so that it makes sense with that structure. So let me erase this here just to get it out of our way. So if we look at this structure and we compare the alcohols that are on the ring here to the alcohols on the ring over here, we put our CH2OH group equatorial. The alcohol that's right next to it is trans. And so with the way that our chair is drawn, this is the up position for that carbon. So that means that the next alcohol in carbon number 4, needs to be in the down position. And it is, because the axial position there would be the up position. And then we can move to the next one. So if we kind of just compare neighbors, these two are also trans. So if this alcohol is down, then this one needs to be up. And it is, because there's our down axial position. And then we continue that trend as we go around the ring. So our next alcohol has to be trans to its previous neighbor. So this one is up, so this one is down. And because this one is down, and this one is also down, so 1 and 2 are cis to each other, then our axial position for carbon number 1 is correct, because 1 and 2 are going to be cis to each other. So really you just kind of have to go around the ring looking at each individual position. So this is the alpha D glucose structure, and almost every alcohol or every substituent is equatorial except for that anomeric alcohol. The beta D glucose does have that alcohol equatorial as well. And if you remember way back when we first talked about chairs, equatorial is the most stable position. So the beta D glucose is the most stable carbohydrate in the chair form. So let's draw this. So we're going to draw beta D idose as a chair. So we have it drawn as a Haworth projection here. So we're going to take this carbon and have it point up, and this carbon and have it point down. So hopefully my drawing here will be okay. That'll work. It's not the prettiest, that's for sure, but it'll work. Okay, so we're going to draw this group equatorial. So sometimes it helps to draw in the axials or see those axials. So I'm going to draw in my hydrogen axial, and then I'll draw in my CH2OH equatorial. And then the next one here is cis to that. So my CH2OH was pointing up. So there is my OH pointing up, and I'm going to draw in that hydrogen. Just sometimes, especially since we haven't done chairs in a while, it helps to see it, both positions. The next one is trans to the previous one. So our last alcohol was pointing up. This alcohol is going to point down. And again, I'm going to draw that hydrogen in there as well. The next one is trans to that neighbor. So it's going to point up because our previous one was pointing down. And there's my equatorial hydrogen. And then my last one is cis to that one as well, cis to its neighbor. So the last one was pointing up. This one's going to point up as well. So it's pointing up as an equatorial position. And there's the axial hydrogen pointing down. So you should be able to take these and turn them into these chair projections. Now, hopefully you saw a little bit of a trend as I went through this. So our CH2OH is always going to be equatorial, which also means because of the position on the ring, it's always going to be pointing up. It's also always going to be pointing up in the Hayworth projection. So as we go around the ring, up, 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 and then we had that one that was pointing down, we can use that as we go around the ring. So we have up, 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 and then that last one that was pointing down over here. It definitely takes a little bit of practice, but really we're just following the trends that are already in that Hayworth projection. All right, we're going to continue with the reactions in this ring, or in this chapter, and we're going to look at the reactions that our carbohydrates go through when they're in the ring form only. So reaction number 8 is creating glycosides. So for our glycosides, that means that we are creating acetals, really. So here is our carbohydrate in its alcohol form. There's our, as opposed to carbonyl form, so there's our anomeric carbon. We're going to react it with an alcohol, so any ROH group here, and that anomeric alcohol turns into an OR group. And this goes from being a hemiacetal to being an acetal. This is what makes a glycoside, and so because of that, this bond right here is called the glycosidic bond. Because we've made a new structure, it does have a new name. So we started with beta-D-glucopyranose because it's in that 6-membered ring. So we're going to change the name. We're going to say ethyl because that's what's attached. That's what our R group was. Beta-D-glucopyranoside. So we went from pyranose to pyranoside because we went to a glucoside. Here's the mechanism for this. Now it looks a little bit crazy, but really it's nothing we haven't seen already. Protonation. When we do that, we create a good leaving group. We made water. So a carbonyl is going to reform and that alcohol is going to leave. You can think of this anomeric carbon as a tetrahedral intermediate because in order to form this reaction, we had to break a carbonyl. So we have these 2 oxygen groups, and we know when we have a tetrahedral intermediate, we either do protonation or we lose a leaving group. So here we lose a leaving group and a carbonyl forms, and then we're going to react it with this alcohol. This carbon, sp2 hybridized, trigonal planar, our alcohol can approach from the top or it can approach from the bottom. And so when it approaches from the top, we're going to make the beta version of the ring. and when it approaches from the bottom, we're going to make the alpha version of the ring. And so we can do either version, and again, we do that attack, that carbonyl opens up, and then we do a little proton transfer to get the hydrogen off of that OR group. And so making that, or attaching that ether group, is what creates a glycoside. Now we can also react our cyclic carbohydrates with a primary amine instead of an alcohol. So this is a very specific example, but we do have a cyclic carbohydrate here, and we have a primary amine. And just like the previous reaction, it could have been a pyranose or a furanose. It wouldn't matter, just as long as it's in cyclic form. And here's our primary amine. So same mechanism, same idea. We're attaching that nitrogen group at carbon number 1, and it can point in the down position or in the up position. And the reason we're specifically looking at this one is because these are the subunits of DNA and RNA. When we have an N-phenyl alpha, or excuse me, the beta version is the part that's in DNA and RNA. So the N-phenyl beta N glycoside. So our ribose in its ring form with that nitrogen attached to carbon number 1. Oh, and then the name change for this one. So the name changes to, so we take off the E from the ribose, and it changes to YL amine, so ilamine, so ribosylamine. Couple more labels. We can label our sugars, our carbohydrates, as either reducing sugars or non-reducing sugars. These are kind of generic labels. It's also something that gets used in biochem, not really organic chemistry. The definition of a reducing sugar is something that can be oxidized, and the definition of a non-reducing sugar is something that can't be oxidized. So we know that if something's oxidized, something else has to be reduced. So if it can't be oxidized, then it's not able to reduce the oxidizing sugar. So that's why it's called a non-reducing sugar, because it's not capable of being a reducing agent. Whereas if it's a reducing sugar, it is capable of being a reducing agent, which means that it can be oxidized. Now, the official definition of these is that a non-reducing sugar is an acetal, and a reducing sugar is anything that's not an acetal, which is a horrible definition. But here's a little bit better of a list. So non-reducing sugars are acetals, which are our glycosides, so same thing here, and that does include the nitrogen-based ones that we just saw. So that means our reducing sugars are the aldehydes and the ketones, so those would be our straight chain carbohydrates, and the hemiacetals, which would be just our regular ring forms of our carbohydrates, so without going through any reactions. Okay, so we have a few slides here to look at what happens when our carbohydrates bond together. So we've been talking about monosaccharides, so our carbohydrates with just one sugar, either in their chain or the ring form. But what happens when they form those rings is that they can react to each other. So we just saw the glycoside reaction. So here's our sugar, here's our anomeric carbon and oxygen, here's a giant R group. It just happens to be another sugar. So this is another type of glycoside, but because it's two sugars that are bonded together, we call it a disaccharide. And so it's when we have two carbohydrates bonded together through that glycosidic bond. Now we do have different types of bonds, of course. So this one is called an alpha 1,4' glycosidic linkage. So let's talk about the language there. So first off, 1,4'. That's because we're using carbon number 1 and carbon number 4 of another ring. The prime is there because it's telling us that carbons 1 and 4 aren't on the same ring. So it's just saying it's a different ring. Alpha is because we have the oxygen on carbon number 1 here is pointing in the down position, which tells us that it's an alpha oxygen. So we have alpha oxygen on carbon number 1 bonded to carbon number 4 on a different ring, and that's what gives us a glycosidic linkage. So I think there's one more thing I want to point out on here for sure is this little squiggly line down here. And so that is telling us that this alcohol could be alpha or beta. So that means that it could be a mix of either one. It could mean that we don't really care. So we've done that with chain forms when we were talking about R and S configurations. So we had a chain like this, and if we had a wedge with an alcohol on it, or we had a dash with a chlorine on it, but we could also have a squiggly line with something attached to it, and that either meant that you didn't know the configuration, or it didn't matter, or it was a mixture of the two. And so this is kind of the case here too. It's just saying that it's not being specified. I think that is it on this one. So this is the alpha 1,4. Here's the beta 1,4. So for this one, it's the same idea in terms of which, sorry, I'm switching my colors there, which carbons are bonding, because it's still a 1,4 prime. So we have carbon number 1 and carbon number 4 of the other ring. But this time it's a beta, so that just means that right here the bond is going to be pointing up instead of pointing down, and that's what gives us a beta 1,4 glycosidic linkage. We can also draw these as Haworth projections, which can definitely be a little bit easier when you're trying to draw these. So we saw the chairs on the last 2 slides. Here are the Haworths. So let's look at the alpha first. We have a 1,4 glycosidic linkage, alpha. So at carbon number 1 here, it's pointing down, and that's what makes it alpha. And yes, it's kind of weird angles, but it is representing that it's pointing down and is going to and from the correct carbons. Here is a beta 1,4 in Haworth projection. So there's carbon 1, carbon 4, and then the bond going from carbon number 1 is pointing up. So it is the beta version. Okay, I'm going to go back up a couple slides because I want to point something out. So we're going to start here. Look at carbon number 4. In every example, because this is not coincidence, this is just how it is, our bond is pointing down at carbon number 4, down at carbon number 4, down, and down. So when we say the alpha and the beta, that's only referring to the direction from carbon number 1, from that alpha carbon. What about our polysaccharides? So now we're looking at having more bonded to each other, and for carbohydrates, poly means anywhere from 10 carbohydrate units all the way up to many thousands of units, just depending on what it is. And we can still have those alpha 1,4 or beta 1,4 glycosidic linkages. That's going to make a straight chain of carbohydrates bonded to each other. Sometimes though they get branches. So this is a alpha 1,6 glycosidic linkage. So notice we're going from carbon 1 to carbon 6 of a different ring, so carbon 6 here, and it's pointing down because it's the alpha bond. So this is creating a branch off of that original chain. So this is really a way to connect 2 chains together. When we have a beta 1,4 glycosidic linkage, it can create intramolecular hydrogen bonds. So here's our oxygen that's part of one ring. Here is our alcohol on the neighboring ring. It can create that hydrogen bonding, which is what can make a structure more rigid. So this is the basic structure of cellulose. And we know that cellulose is found in plants and in grass. And we also know that we can't digest that. And one of the reasons why is because that structure is so rigid and so stable. And so grazing animals like cows and horses do have enzymes that can help them break those structures down, but it's something that we don't have. So when we have that beta structure, we get those intramolecular bonds holding it in that very rigid shape. So this is our cellulose basic shape. And when we have the alpha bonds, it folds itself into the helix shape. And we do still have the intramolecular bonds, but they're holding it in a way into that helix shape, and so it makes it more water soluble. It also can make it more stretchy, and so when we have starches that are stretchier like cotton, it's because they are held in that helix shape, so it gives it some flexibility. All right, couple more slides. So we can also have some other variations of our carbohydrates. So the first ones are deoxy sugars. So you've probably heard that term before because we know that DNA is deoxyribose nucleic acid. The deoxy is because one of the oxygen groups has been removed. It's been deoxygenated. And so for DNA, it's the D form of ribose in a 5-membered ring, and carbon number 2 has had its alcohol group removed. So D2-deoxyribose is the sugar component of DNA, and RNA, ribonucleic acid, doesn't have its sugar, doesn't have a derivative of the sugar. It's just the ribose in its ring form. So that alcohol on carbon number 2 is still there. So that's the big difference between the carbohydrate portions of DNA and RNA, is whether or not that alcohol group is there. And then the other one that is naturally occurring are amino sugars. And so with an amino sugar, one or more of the alcohols has been replaced with an amino group, with an NH2 group. And so this one in particular is a naturally occurring antibiotic. We also find amino sugars in bacterial walls. Sugar types are what determine what type of blood type you have. And so we have oligosaccharides, so this is somewhere between 3 and 10 carbohydrates that are attached to the oxygens or the nitrogens on the cell walls of red blood cells. And so if you have type A or type B or type O blood, these are the specific sugars that are going to be attached to your blood cells. You might have heard that type O is the universal donor. This can show you why, because the part that's in red here is identical to types A and B, and the only thing that's different is this last sugar on these two. Type O doesn't have that last sugar, so it is compatible with the other blood types. So attached to those cell walls are these different sugars that identify us as different blood types. All right, last topic is looking at artificial sweeteners. So we know carbohydrates are different types of sugars depending on where they're found. We also know that there are a lot of artificial sweeteners out there. One of the one that's more new is called Splenda. Its more chemical name is sucralose, and sucralose is made from sucrose. The difference though is that it has chlorines on it. So 4 of the alcohol groups have been replaced by chlorines. And so our body still is able to recognize that as something that tastes sweet. So the way that it hits your tongue, it still tastes sweet. But because it has these chlorines on it, our bodies don't recognize it as a carbohydrate. So it just gets eliminated from your system before your body has a chance to metabolize it and absorb it. All right, that is it for the rest of this chapter. So definitely the big takeaway from this is being able to draw the carbohydrates as alpha and beta, pyranose and furanose rings, and as chair projections. And then we did have a couple reactions in there, but I think the rings are going to be the ones that require the most practice. And that is it, though. Thank you guys for being so flexible and understanding. I appreciate it.